Previously on Weiss Guy Science.
What does DNA actually do for cells? Eye color is determined at conception when mom and dad's chromosomes align to make you the zygote. The pathway that I just described is called the central dogma of molecular biology and proteins. Therefore, there are three stages of transcription: initiation, elongation, and termination.
Sleep, wake, sleep.
Justin!
What was that? Hold on. Oh my God! What are you doing? Whew, sorry about that. It was just a cat knocking something down. It's all good. In the previous video, I discussed transcription of a specific sequence of DNA into messenger RNA. The example I used was melanin beginning to darken a 2-month-old baby's eyes. When messenger RNA is made in a nucleus, it leaves via nuclear pores and heads to a ribosome. Ribosomes function in making proteins and can be found either on the rough endoplasmic reticulum just outside of the nucleus or within the cytoplasm of the cell. Proteins produced and destined to be secreted out of the cell are made on the ribosome of the rough ER. Proteins made within the cell are made on ribosomes in the cytoplasm. So why are proteins so important? They are highly versatile, ranging from structural components of cells such as cytoskeletal filaments to functional proteins such as insulin to help maintain blood glucose levels after a meal. Some proteins are retrieved from our diet, but any protein made by the body derives from DNA and are copied onto mRNA and then translated into a series of amino acids, which are then used to build these very proteins. How does protein synthesis work? In order to understand this, you need to look at the machinery behind this process. A ribosome is composed of two ribosomal subunits, large and small. Both subunits are made of a series of peptides and ribosomal RNA. Our RNA is made by these nucleolus of the cell, which eventually builds ribosomes for the cell. The small subunit's job is to bind to the mRNA to be translated, while the large assists the tRNA or transfer RNA to bind to the mRNA and deliver the appropriate amino acid. Essentially, tRNA acts as an adapter, where they connect the mRNA and translate the language of RNA, which are nucleotides, to the language of amino acids. However, each tRNA can only bind to a specific sequence of mRNA, and thus over 40 to 60 different types of tRNA exist in the cytoplasm. You may also notice that there is a 3-letter sequence at the base of each tRNA. This is called the anticodon, and its job is to bind with the codon on mRNA to ensure the correct amino acids has been sent to the ribosome from tRNA. So how does tRNA know to read the mRNA and deliver the appropriate amino acid? RNA contains 4 nucleotides, A, C, G, and U. The ribosome presents 3 nucleotide bases at a time called codons. And since there are only 4 types of nucleotide bases and all codons are read in groups of 3 nucleotides, there are 64 codons that exist. And of this, 61 codons translate into one of 20 amino acids and 3 translate into stop codons. Before discussing stop codons, notice that one particular codon labeled AUG. This translates into an amino acid called methionine, which is also known as the start codon. The purpose of the start codon is to signal to the ribosome to begin reading the mRNA and ordering the appropriate tRNA to deliver the appropriate amino acid. The stop codon signals to the ribosome to stop reading the mRNA and disconnect from it. So, how do you read this chart? Though it may look a little intimidating at first, it's actually really easy. You first start by taking the first letter of the codon, which is on the left side of the chart, and then find the column on the second letter, and finally go to the right of the chart and find the third letter of the codon. For example, if you need to translate AGG, you will go to the third row, last column, and line up with the last G. So AGG is
Arginine, which coincidentally is the pirate's favorite amino acid.
Another version of the genetic code is a circular chart, which I find much easier to read. You start in the middle and work your way out, and once again,
You find me amino acid to be arginine from the codon AGG.
and this relationship between codons and their amino acid is referred to as the genetic code. And although it looks a bit, well, boring to look at, of course, you really can't undervalue it. One of the main ideas behind evolution is that all life has evolved from a common ancestor. And this genetic code is one of the strongest pieces of evidence for that universal relationship. Why? Well, with very few exceptions, the codon GUA is valine, whether you are a frog, an amoeba, a staphylococcus bacterium, or even an oak tree. Finally, the genetic code is redundant, meaning that many amino acids have more than one codon for them, and this is one way in which a mutation can cause no effect on an organism. So if CCU, which translates into proline, were to mutate into CCC, it's still proline, therefore not changing the amino acid structure of the protein in question. Protein synthesis occurs by a ribosome reading mRNA to order tRNAs to deliver amino acids. Ribosomes specifically are there to build peptide bonds. A peptide bond is formed when two amino acids bond together, and this occurs when the amino end, or NH2 or NH3 end, and a carboxyl end remove a water molecule, allowing for the two amino acids to form together. Protein synthesis occurs when a ribosome in a tRNA translates the codon on mRNA and a correct amino acid is delivered to the ribosome. Just like in transcription, there are three stages: initiation, elongation, and termination. In initiation, the small subunit of the ribosome binds to the mRNA, and the mRNA moves down the track until it is met with an AUG codon. From there, a tRNA with the anticodon UAC and amino acid methionine is delivered to the ribosome, and the large subunit binds to the small subunit to begin translation. Notice that it is in the compartment labeled A. This is the site where tRNAs enter the ribosome. Next, the tRNA moves to the P site where a peptide bond will be formed when the next amino acid arrives. By the time the tRNA reaches the E or exit site, it is no longer bound to the amino acid and exits the ribosome. After initiation, elongation begins with methionine at the P site, and another tRNA with the anticodon AGC matches with the second codon UCG on mRNA. And this provides the ribosome with
serine.
Cysteine is then moved to the P site where a peptide bond forms between methionine and cysteine, and the empty tRNA transfers to the E site where it exits the ribosome. And this process continues to repeat with the next mRNA codon UUU, which will bring about a tRNA with the anticodon AAA with the amino acid phenylalanine attached. The tRNA will move to the P site where another peptide bond is formed between phenylalanine and a growing chain of amino acids. tRNA then leaves via the E site. And this pattern will continue to repeat until the ribosome encounters a stop codon. And there are three mRNA codons that do this: UAA, UGA, and UAG. Once encountered, termination begins where no amino acid is delivered, but the ribosome simply detaches from the mRNA and protein synthesis concludes. Afterwards, the proteins will be folded into specific shapes and used in biochemical reactions, gene expression, and signaling for the organism. In the example of melanin darkening a baby's eyes to brown, these melanin proteins would then be expressed in the baby as brown eyes. Of course, changes can occur within DNA, especially in the development of egg and sperm cells, and these changes are called mutations. While mutations get a bad rap, as some can be deadly, and many are, well, harmless and do not affect an organism, it is rare that sometimes a mutation can occur which introduces new and beneficial genetic features. Here, I will introduce some examples of simple point mutations. And please keep in mind that more complicated mutations can occur with entire sections of chromosomes or multiple nucleotides changing. But for the purposes of this video, I will only cover point mutations. Point mutations occur when a single nucleotide base changes. The first type of point mutation is called a silent mutation. Remember the redundancy of the genetic code? This occurs when a mutation occurs, but the amino acid remains the same. So even though the nucleotide mutated, it did not change the amino acid, and thus the protein made will remain the same. So both GGG and GGU encode for glycine. However, it is possible that a mutation that changes the nucleotide can change the amino acid. In this case, GGG mutated into GUG, and as a result, what should have been glycine is now valine. While this doesn't seem like that big of a deal, a missense mutation could affect the organism negatively or positively. A good example is in North America, many mice are brown in color to help camouflage. Brown fur coats are a result of protein synthesis for that fur color. However, a missense mutation in this species had led to a subspecies of field mice to be more tan in color to camouflage well in sandy conditions such as beaches. So they're called beach mice. The other type of point mutation is called a nonsense mutation. Here, a nucleotide is converted into making a stop codon, prematurely shutting down translation. One example might be a baby born with cystic fibrosis, and this disease causes an overproduction of mucus in the lungs of a person and is usually deadly. Mutations are important to evolution as they are the only mechanism that can introduce a new gene to a population. In other words, mutations may lead to the formation of new adaptations or even new species. DNA is the great unifier of life, as all life has DNA and utilizes the same genetic code. But how did this molecule result in the millions of species that exist on this planet today and in the past? How does life evolve over time? Well, in our next set of videos, we will be looking at evolution.